In a typical stress response, there is a release of T helper 2 cytokines, which are pro-allergenic and are involved with a very robust inflammatory response. However, it has been observed that during repetitive stress exposure, during allergic sensitization, that there is a T helper 1 cytokine bias response, which is still involving inflammation but is more confined. It has also been observed that there is the release of substance P during repetitive stress exposure. Substance P is released from nerve fibers and signals through the dendritic cells, which then in turn activate T cells to differentiate into either T helper 1 cells or Th2 cells. In this experiment, they looked at the effects of repetitive stress exposure causing the release of substance P on allergic sensitization. They did this by creating AID, or atopic dermatitis-like allergic inflammation models. They did this by injecting mice with chicken ovalbumin and aluminum hydroxide. The aluminum hydroxide was used as an adjuvant to cause the slow release of the chicken antigen. After this first exposure, they then caused an allergic challenge by injecting the mice with chicken ovalbumin again. In addition to AID models, there was also a stress plus AID model, and in these stress mice models, the mice were exposed to a sound irritant every 4 seconds over a 24-hour period. Then after the stress exposure, the mice were injected with the allergic challenge of chicken ovalbumin again. It was found that in stress mice, there was increased interactions between nerve fibers and dendritic cells, which signifies the substance P signaling. In addition, it was found that more dendritic cells were matured in stress plus AID models. This was indicated through increased expression of CD80 and CD86 co-stimulatory molecules. These molecules are involved with the activation of T cells, in addition to adhesion factors like VLA4 and LFA1, which are also increased in stress plus AID models. In addition to dendritic cell maturation, there was also the increased skewing of Th1 cell response as opposed to Th2 cell response in stress plus AID models. Stress plus AID models increased expression of interferon gamma and tumor necrosis factor alpha, which are Th1 cytokines, as opposed to the AID group, which had interleukin 4 and interleukin 5 increased expression, which are part of Th2. In addition to the different Th cells response, there was the increased expression of CD25 expressing T regulatory cells in stress plus AID models. T regulatory cells are involved with the immune suppressing response. So this was very important in the stress plus AID models, which had very significant increases of these cell quantities. In addition to the T regulatory cells, there was increased expression of the surface marker CD25, which is a receptor for IL-2 or interleukin 2. Interleukin 2 was also increased in stress plus AID models as opposed to AID only models. Interleukin 2 is actually involved with T regulatory cell proliferation, so this enhanced the effect of the Th1 cytokines. Finally, there was reduction of allergic inflammation on site of the chicken antigen injections in stress plus AID models. It was found that there was less eosinophil migration in stress plus AID models as opposed to AID, because eosinophils are involved with releasing histamine granules, involved with causing an allergic reaction. There was also less epidermal thickening in stress plus AID models. In conclusion, it was found that substance P, which is released during repetitive stress exposure, was responsible for quelling the immune allergic response in stress plus AID models. This was found through increased contacts between nerve fibers and dendritic cells, in addition to dendritic cell maturation, Th1 cytokine bias release, increased T regulatory cells, and also interleukin 2, and decreased inflammation on site of the chicken ovalbumin injection.